Okay, thanks for the organizer for inviting me. Okay, I changed my title because it was a little bit too much. First, I changed it, I insert the word towards because we are not there yet. And second, I skipped the anatomy because I would not have been able to cover all the topics I want to cover. So the current state, most of the studies we do with MRI are still on the group level, and they have provided tremendous insights. And still to this day, a lot of even the common diseases, we don't know sort of the brain correlates and so on. So therefore, group analysis will continue and will continue to provide interesting insights. In addition, there's sort of a pressure to go to single subject level. For example, if you do neurosurgery, we don't want to know on the group level where we should target, but we want to know on a single subject level where we want to target, for example, deep brain stimulation. So we want to link behavior and brain features, now brain decoding or brain reading. But for clinical, we want to identify patients before they develop the diseases who are vulnerable, and we can perhaps intervene. We want to do diagnosis, prognosis, and tailored therapy. So imagine a case, we have a drug trial, and this drug works on 10% of the patients, and in 30% of the patients, it has negative effects. So we would think this drug did not work, or we should not continue with this. But imagine we would have brain biomarkers which would identify these 10% of the patients before we try the drug. Then this drug would actually be extremely successful. So if we therefore can identify or subdivide patient population based on brain features and not based on behavioral symptoms, then we might be actually more successful with many interventions compared to what we are now. Then we can talk about anatomy. Obviously, we had in the 19th century phrenology, patient HM, we have the brain cognitive ontology, prescribing different function to different brain areas. And then we have in the last 2 decades or 1.5 decades, lesion network mapping, where we don't look just at lesions at one location, but we look into and watch networks they're integrated. You can think of this as like a spatial Taylor expansion to 0th order and 1st order. Then in functional, we can also have the same thing. The dominating approach in functional MRI is sort of looking at activations, but more and more connectivity-based analysis is also prevalent. And then we have the integrative approaches, what will be discussed later by Petra on the virtual brain, the digital brain, the digital twin idea with Katrin Amunds. We have seen yesterday a talk on the foundation models and precision MRI. So what is needed to go to single subject MRI? One is we need good data. So it's paradoxically, in order to do single subject MRI well, we have to go to large data sets. And what does large data set mean? We can either go to, I don't know if I have mouse, okay. We can either go to a large N, large number of subjects. Okay, let's see. Large number of subjects. And or we go to low number of subjects, but extensive amount of scanning. So the precision MRI, what Boris has shown yesterday. And both have their usefulness. And the reason why we need big data is we want to, in order to understand individual specific brain patterns, we have to understand variability. We have to understand inter- and intra-subject variability. And we have to understand what of this variability is actually meaningful and what is sort of considered from a certain research question noise. And therefore, we need the big data in order to be able to develop this kind of models of variability. Then we have to also ask how we acquire data. For example, in fMRI, do we acquire resting state, which is the most popular approach, or task-based? And I think naturalistic stimuli have a big future. What is often neglected are generative models. We need also models of linking the biology to the observations. So for example, in fMRI, which I will talk about, what we are measuring is actually an indirect signal of neuroactivity. We are interested in neuroactivity, but we are measuring something on the blood level, and what can happen, what can go wrong, and how do we understand neuroactivity from fMRI. The same can be done also on the anatomy level. We have seen yesterday from Alan a talk on free water. How is free water actually linked to the biological processes going on in the brain? And what's actually sort of what does it mean? So we need observation generative models to link what we are measuring to biological parameters. And then this will help us to separate signal from noise. Then we need feature extraction, because in an ideal world where we have infinite data, we can let AI do everything. We don't need to think about the features, because we just feed everything into a neural network, and then it extracts all the features which are relevant. However, if we have limited data, and especially in disease, we will have always limited data, then we need to extract features in a guided way, and then do some sophisticated analysis. So feature extraction will be an important step. And we need a normative modeling, what's group and what's individual specific. So in my talk, I will not cover everything. I will talk about task fMRI, then generative model linking fMRI signal to neuroactivity, then how we can use this to have insights into diseases. Okay, so the generative model I'm talking about is in the framework of dynamic causal modeling. It doesn't have to be. It's essentially a model of the hemodynamic signal. I will show you a few slides on the theory. If you cannot follow the theory, that's okay, because I will come to the applications afterwards. So imagine you have a stimulus, like a video, like a movie. You scan the person, and then you have activation in different brain areas, V1, MT, FFA, and so on. So in each of the areas, you have time courses. You can do general linear model analysis and get activation maps over the brain. Or you can relate the time course to each other to get connectivity between the brain areas during the task. Usually, when people do this kind of analysis, they analyze on the level of the Bolt response. The Bolt response, Bolt stands for blood oxygenation level dependent. And often this works well. However, the Bolt response is a signal from the blood. And I will show you an example why it can go wrong. So if you want to understand it, we have to go to neural level. On the neural level, we have synapses, we have spikes, we have neurotransmitter release, we have astrocytes. We simplify this into just a time course of neural response. Okay, and then this neural response leads to blood flow change, and this process is called neurovascular coupling. The blood flow changes blood volume and deoxygenated hemoglobin. Deoxygenated hemoglobin is paramagnetic, and therefore we measure this as a Bolt signal. So if you look at this chain of events, the Bolt signal is quite an indirect response of neuroactivity. So every step in between where something happens, which we do not account for, can lead to a misleading picture of the neuroactivity we want to actually measure. So if you want to therefore develop a model of the Bolt signal, we have to develop a model for neuroactivity, on neurovascular coupling, of hemodynamics, and then the physics, how the deoxygenated hemoglobin is translated in the Bolt response. This model is quite complex, and therefore a lot of people are afraid to use it because they say, what if the model is wrong? And this is a fair argument, but not using a model is also a model. So and not using an explicit model, you make an implicit assumption that all the variance observed in your time course is actually a variance of interest. And which means everything what we see in the Bolt signal is actually reflective of neuroactivity, which means essentially you're assuming an identity transformation, which also is a model. So therefore, using a model which might be wrong is not necessarily, should not prevent you to use a model. I want to show you just one example, which has been published 2 weeks ago on psychedelics. So as you know, psilocybin is a drug which has been very popular in neuropsychiatry in the last few years. And this paper published in Nature Neuroscience, they say our results demonstrate a dissociation between drug-induced neuronal and hemodynamic signals, indicating a need to consider neurovascular coupling effects of psychedelics when interpreting blood-based measures of brain function. So what they found is when they used this drug, that actually the hemodynamics and neuroactivity were influenced, but they were influenced in different ways in different brain areas, which means the coupling between the neurons and the bolt signal was affected, but in a region-specific way, which means if you would do, say, resting state analysis with or without the drug, you actually get wrong answers because at least that's the claim of this paper, what you are measuring is not the changes in neuroactivity due to the drug, but changes of the neurovascular coupling. So therefore, by ignoring this chain of events, sometimes it might go well, but this paper argues it might go wrong, and especially in many drugs what are used in the clinic, they have both neuronal and vascular effects. Okay, now I will not go through the equations, but I just present you how this model is structured. So on the neural level, we have excitation and inhibition, and there's an adaptive link between these two, and extraterritorial neurons from one brain area is connected to extraterritorial neurons to another brain area. Then the neurons, when they're active, they release vasoactive substances, and they change blood flow. And they change it in a feed-forward way, which means when the neurons are active, they consume energy. They just say we need more blood flow to deliver us more oxygen and glucose, and therefore they change regardless how much there is already available. Then we have the hemodynamics, which is the balloon model from the 1990s. So in summary, on the neural level, we have adaptive inhibition excitation, neurovascular coupling is feed-forward, vascular dynamics, blood flow and blood volume uncoupled, and so on. I want to show this sort of the model we have developed. This is described in these papers. The standard model, which is more widely used from Friston from 2003, it has different structure. They don't have excitation inhibition, they have only excitation. They have a feedback loop between blood flow and the vasoactive signal, and the balloon effect is sort of removed. And we have compared these two models widely. The most important thing for sort of users is, depending on the model, you get different connectivity parameters, which means if you use sort of a different model, the connectivity between brain areas the calculated connectivity between brain areas are different, which means because if part of the data the variance of data is not explained, then it's explained as connectivity between brain regions. And therefore, using the right model will give you, or using a different model will give you different connectivity parameters between brain areas. I want to show you two examples how we have applied it into disease populations. So how we do this? So we have an input stimulus, we extract fMRI activations in different regions of interest, then we do dynamic causal modeling analysis, and then we get a connectivity between region 2 to region 1, region 3 to region 1, region 3 to region 2. So we get different connectivity parameters. We do the same for across subjects, and then we can then use these connectivity parameters and then do some feature analysis, clustering, or machine learning, and so on. This we do while we don't say to the algorithm which one was patient, which one was health subject. We just pull all the connectivity parameters between subjects together and then see whether the connectivity parameters between the subject cluster in a way that is meaningful. And then the second parameter we have is how do the connectivity parameters, or how do the clusters based on connectivity parameters in each subject differ, which means, for example, if a subject is in between two different clusters, say control and patient, and you cannot differentiate, then the index would be 0.5. If it's 1, then it means it's very clear in the one or the other cluster. So the data is on major depressive disorder. This was an experiment where they listened to music and pure tones for 30 seconds, positive music, positive connotative music, pure tones, negative music, and so on. This was around 40 subjects, 20 controls, and 20 major depressive disorder. Then we have a connectivity graph with different connectivity, for example, orbitofrontal cortex to dorsolateral prefrontal cortex with feedback. And then for each subject, we get a connectivity parameter for each of the brain areas. And this is shown here we have in total 16 connectivity parameters. What you see here is sort of the average connectivity, and the arrow bars are the connectivity difference between subjects. Which means, for example, if you go to number 10, or let's say number 11, that's the connectivity between orbitofrontal cortex and dorsolateral prefrontal cortex. And the arrow bar, it means there's some connectivity, high connectivity between these brain areas during the music listening. And then you have an arrow bar which shows how much it varies across subjects. So now this is a very important step. Usually when people analyze connectivity, what they take is the strongest connectivity parameters between brain areas. And I think this is a wrong idea. For example, if you take 7, you have a high connectivity parameter and low variance across subject. But this means that connectivity parameter is actually non-informative about subject identity. So what is important, for example, if you go to number 2, the average connectivity was 0, but you have a high variability. So, which means if you do feature selection based on connectivity, what you have to use is not the strength of connectivity, but the variance across subjects in order to be able to differentiate between subjects. So therefore we selected the ones with the highest variability. If you do this, then, as I said, the algorithm does not know who is a control subject, who is a patient. And what you find is actually we have a classification accuracy of 95%, which means just based on the brain parameters alone, we could say with 95% confidence who was a patient and who was a healthy control, which is sort of quite impressive. When you look at then the standard DCM from Carl Friston, it's also quite good. It's, however, a little bit lower, is around 80%. Now, the important finding is if you look at activations, so if you look at the brain activations during the music listening task, the accuracy is just barely above chance, which means the controls versus health subjects, if you do GLM, the difference between these two populations is almost not existent, which means they activate on average the same way, so depressed patients versus healthy controls, but the connectivity actually informs us about the identity of the subject, and even though the activation itself did not inform us. So that's a quite important finding, showing that connectivity actually provides us information which activation does not do. So this is the cluster index. If it's above 0.5, it was control subject. If it was below, it was a depressed patient. And you see most of them, controls are above 0.5. Only one subject was, subject number 14, was somewhere in the middle. And the same with depression. So that's where the 95% classification accuracy comes from. Now, I mentioned if you take the strongest connectivity parameters, so the top 8 strongest connectivity parameters, what you find is actually we cannot differentiate between controls and patients, which means the strength of if you do group analysis and you choose the brain areas which are most connected during the task, they might actually not be informative. So feature selection is therefore very important, and so therefore you have to select based on variance and not based on the strength itself. And here, again, you see the activation does not inform us about subject identity. Now, how is this useful for clinical? So now that we know that we can separate controls from depressed patient based on connectivity parameters, we can look now on the average connectivity difference between these two populations. And this is shown here. So it's controls minus depressed patients. For example, if you go to this one, I don't know, you cannot see it. So the third one from the left, it's negative, which means depressed patients have higher connectivity in between dorsolateral prefrontal cortex and ACC, which means there's a stronger connection between these two brain areas. So if you would do intervention, for example, using TMS or electrostimulation, and if we would inhibit this connection, this might be a way to reduce depression, for example, because they express too high connectivity between these brain areas. In addition, what we have done is we tried to find whether we can actually find subpopulation between depressed patients. We were successful. However, because sample size is not big, we don't make a big claim out of it. Now I want to show you what we are doing right now. We do MR-guided focused ultrasound, which means, for those of you who don't know, that's MR-guided focused ultrasound is a helmet in the MRI scanner, and then the focused ultrasound sends ultrasound waves to a certain part of the brain, heats that part of the brain in order to do a lesion. And the idea is if there's a malfunctioning connectivity between two brain areas in a certain patient population, lesioning a part of the brain will actually improve their symptoms. And this is done at Sunnybrook, and there they do MR-guided focused ultrasound lesioning OCD. And here you see in the circle and the dark spot are actually the lesions, where they lesion the ALIC, anterior limb of the internal capsule, bilaterally. And what they see is that these were treatment-resistant OCD patients, which means they have tried every type of treatment and nothing has worked in these OCD patients. However, 2/3 of the patients, after this lesioning, they get better. And they do longitudinal assessment in order to see whether they develop any negative symptoms. But so far, they don't. And so it's quite a big success. Now, and these are two different patients. And one patient got much better than the other patient. And now the question is, the responders versus non-responders to this kind of therapy, can we, before the surgery happens, can we differentiate them based on their connectivity values, who will be a responder and who will not be a responder? So based on this idea that if we determine the connectivity parameters, how they differ from control subject, if we know where the connectivity most deviates from healthy controls, if we then reduce or increase that connectivity to improve the symptoms of the patient. In a second study on anxiety disorder, that's also around 40 patients, this now with faces, angry faces versus fearful faces. We have a different connectivity graph, and we get the same result. We get around 90% classification of who had anxiety disorder, who was control subjects, just based on connectivity alone, without using behavioral data. Again, the standard DCM is a little bit less effective, and again, the same finding that activation actually was not informative to differentiate anxiety disorder patients from control subjects. I hope I was not too fast. So the summary of this part is unsupervised clustering alongside feature selection based on variance with hemodynamic modeling, effective connectivity features can accurately differentiate patients from controls solely based on task fMRI data. I also think it will be also possible with resting state data, but we have not applied to resting state yet. This is not achieved by activation-based features and also less effectively with standard DCM. I now switch to a different topic, to high-resolution fMRI. I don't know if anybody has a question at this moment. Otherwise, I just continue. So high-resolution fMRI, if you look at the cortex, what already Katrin has shown, we have sort of different layers. Layer 1 to 6, so top is the CSF and bottom is white matter. And then usually this is studied with laminar probes, measuring electrophysiologic activity across layers. And there's a microcircuit model which says that input goes to the middle layers, output and cortical input to top layers, and feedback and output to thalamus in the bottom layers. Now, The thickness, I hope I don't say anything very wrong, is between 2 and 4 millimeters, and which means if you overlay a standard voxel, fMRI voxel, we are sort of covering the entire cortex, so we can say where something happens, but we can actually not say in which layer something happens. So we cannot really look into the processing stream of the cortex, but we can just say there is something happening in that part of the brain. However, if you go to a higher resolution fMRI, where, for example, we go to 0.7 millimeter isotropic voxels, and actually now people are trying 0.5, which compared to histology is very bad, but non-invasively is very good. So then we can actually see different depth of the cortex, and we might be able to differentiate between output, input, and feedback. Now, one of the consistent observations is in electrophysiology, we get this from a study from Alexander Meyer, where he has shown different cortical activity in different cortical depth. So top again would be CSF, and bottom would be white matter, and x-axis is time. In fMRI, we have usually on the left white matter, on the right CSF. You can already see that the lamina activation fMRI looks very different from electrophysiology. So there's a problem. The electrophysiology and fMRI lamina depth profiles are looking very different, not how we expect. And this observation that the signal increase from white matter to CSF is observed in almost every study. So if you look at this, then the conclusion might be we are not able to measure with fMRI cortical depth activity, even if you have the spatial resolution for it. And the reason for this is venous artifact. This is a low resolution study, but I like this study because they show how even in standard resolution fMRI, something can go wrong. So this is a study where you see amygdala activation and visual activation. If you go to the next slides, what you see is if you follow where the arrows are through the slices, what you actually see is it's actually a vein. It's a draining vein from the visual cortex passing through close to the amygdala. So therefore, it's not necessarily activation of amygdala, but it's a vein which drains the deoxygenated hemoglobin away from the visual cortex and away from the brain. But because it passes close to the amygdala, people might mistake this draining vein effect with amygdala activation. And there are certain brain areas which are known. Yeah, there are certain brain areas which are known where a big vein passes, which are difficult to study with fMRI. For example, V4 is one of these brain areas where V4 activation is most likely not V4 activation, but a draining vein passing through V4. Now, if we go to higher spatial resolution, then we have the same problem. So on the left, so on the top, you see again the surface of the brain. On the bottom is the white matter. Blood is delivered via the arteries, goes through the capillary bed, then it's then drained to the veins and goes up. Now imagine you have activation layer 4, neuroactivation, deoxyhemoglobin is produced, then this deoxyhemoglobin is drained to the surface, which means in layer 3 there will be a change in deoxygenated hemoglobin, in layer 2 there will be a change in layer 1 and in the surface, which means even though neuroactivity was in layer 4, in fMRI you will measure also activation in layer 3, 2 and 1 because there's a change in deoxygenated hemoglobin. So this you can model relatively easily using mass balance. So you have deoxygenated hemoglobin volume in each layer, and then this drains into the vein, and then it's drained to the surface. Now, with modeling, you can then estimate the effect of one layer activation versus another one. If you look at the light blue one, we had induced activation layer 6, but and the draining vein effect also generates activation layer 5, 4, and so on. If we know the effect size, we can actually then calculate it out. So we have measured with artificial spin labeling, high resolution study. So on the right, you see blood flow, and on the left, you see the Bolt signal measured. Again, the Bolt signal increased from white matter to CSF. If we insert the blood flow reflects more or less the neuroactivity profile. if you insert into the model, what you actually see, this layer profile of blood flow predicts accurately the bolt signal, which means we understand that why the bolt signal has to have a different layer profile than the electrophysiology. And by understanding it, we can actually then sort of remove this effect. So we have done this in one study. So this is V1 and V2 at different conditions. Again, you see from white matter to CSF, increasing V1 on top, V2 on the bottom. After applying this model, what you find is actually, for example, in V4, we get a peak in layer 4, which we expect for feed-forward stimulation. In V1, we actually get a peak in layer 6 and layer 4, which is actually electrophysiologically confirmed, where for feed-forward stimulation, there is a direct input to layer 6 from LGN. So summary of the layer fMRI model. So we observe a discrepancy between high-resolution fMRI spatial profile and laminar electrophysiology, and this can be explained by the ascending vein effect of the bolt signal, and the layer hemodynamic modeling capture this effect and can successfully remove the spatial bias. So if we go, now how do we continue from here? So we have the neural level, where we have feed-forward, feedback, lateral connections. We have the vascular level, and we have then the voxel level of fMRI. This B and C we have, I think, very well under control, but now let's look at the neural level. And there, Katrin already has shown that the layer distribution of cells, and they have shown that if you look at different brain areas, the cellular distribution actually varies, which means also when we look at the receptor architecture, that they vary across the cortex. So there's no reason to assume that the cortical circuitry should be the same in every brain area. However, we have no model of the cortical circuitry at this moment, at least in humans. So the canonical microcircuit model from Douglas and Martin, which says input goes to layer 4, interactions layer 3 and 2, and feedback layer 5 and 6. So this is sort of illustrated in this picture. This was developed in the cat visual cortex. And so therefore, we actually do not know how the circuitry is in the different brain areas. So using this model, we can actually sort of test this. So we have the current model where each layer has excitation inhibition, but they're not connected to each other. We can have a canonical Merkel circuit in one region, or we can connect the different regions with each other. And we have an ongoing project. We don't know how much ongoing it will be because it's NIH funded, where we have epilepsy patients where during neurosurgery, we are planning to do SEEG and also laminar electrophysiology in order to develop a neural model of electrophysiology, invasive electrophysiology, use then the model to calculate the bolt signal and measure in the same patients before surgery using the same task, the laminar profile, and then being able to actually see how well the neural model derived from electrophysiology corresponds to the fMRI model or the fMRI signal measured. To summarize this part, laminar fMRI model integrated with models of canonical microcircuit can translate invasive laminar electrophysiology to high-resolution fMRI. And model inversion can help infer local neuroactivity between cortical layers and connectivity between different brain areas in different layers. At the end of my talk, I want to just mention how this might be linked to the big brain or in general histology and psychoarchitecture, receptor architecture. I mentioned that different brain areas have different circuitry. For example, circuitry model for hippocampus looks like this. Emily Finn, in their laminar fMRI paper in 2019, they had a microcircuitry model for the prefrontal cortex, and they used this to predict the fMRI signal. However, these are very speculative because we don't really know what the microcircuitry models in different brain areas are. So the basic idea would be to use big brain, but also other type of data like psycho architecture and receptor architecture, then data like from Bradslav's lab about neurotransmitter and neuromodulator distribution. There's a reason why the neurotransmitters and neuromodulators are distributed unevenly across the cortex, because the circuitry is different. Now, can we reverse that logic and say, can we use this information to actually develop heuristically microcircuits across different brain areas? There was a paper, I don't know what Alan thinks about that paper, but they claim that based on big brain, they can generate like neural distribution and microcircuit model in the hippocampus. Anyway, this sort of is a research program where we essentially take many different type of data, high resolution data together to link neural distributions, soma distributions, receptor architecture, and many other data to link to microcircuits to predict electrophysiology, which then can predict the fMRI signal. So if we have then a brain area specific microcircuit model, then using the model what I have shown about laminar fMRI, then we can actually in a forward way predict how input to a brain area will propagate to the fMRI signal. Then having real fMRI signal measured, we can actually do Bayesian model inversion and determine the parameters of the microcircuit. And then tying it back to single subject MRI, if we have laminar connectivity and laminar neuroparameters, then we can use them as single subject classification. For example, if somebody has hallucinations, it will be different than a sensory stimulation distribution of laminar activity, because hallucination, if it is top-down from a higher brain area to a low brain area, it will have a different laminar distribution than a feed-forward sensory stimulation. So based on in standard resolution fMRI, we might not be able to differentiate these two type of activation, but having laminar neuroparameters and laminar connectivity, you might be able to have better differentiation between subjects. To summarize, to come to the end of my talk, so single subject MRI is still early in development. There have been many impressive papers, but I think we are still very, very early in the approaches. The best approach will likely depend on the research and clinical question. We have to think about the data, what data is the most appropriate, because if the data does not contain the information which allows you to differentiate between subjects, it will not be useful. We need models. We need observation models, how to link the biological parameters to the measured parameters. We need a feature extraction method, because we will have limited data and we cannot learn everything from the data itself. And it will depend, all these things will depend on the application. I have today showed task-based fMRI, but the approaches can also be applied to resting state or naturalistic stimuli. I talked about the generative biophysical model of fMRI signal. In our case, connectivity was very valuable, much more valuable than activation itself. But the feature selection is a very important, crucial step. You have to use variance and not the mean connectivity strength across subjects. And then we found that we can, without using behavioral data, differentiate between patients and control subjects. And we even tentatively could, within the patients, find subdivisions. So we could actually show, at least in the limited sample size, that depressed patients cluster in two different clusters, so that they're actually two different diseases. And what I mentioned, if we apply therapy and it works on 50% of the patients, if they are in fact two different diseases, it might actually work 100% of the patient category 1 and 0% on the patient category 2, by using then connectivity as differentiator between possible responders and non-responders, might actually make a big leap in the development of therapies. In the second part of my talk, I talked about high-resolution fMRI. And fMRI is a very imperfect tool compared to a lot of the things we have seen by Katrin and Alan. But it's the best non-invasive tool we have. So therefore, we have to link the rich data which we can obtain with psychoarchitecture and receptor architecture, and link it to non-invasive measures to be able to use this in a non-invasive way. So their modeling will be important. and we need microcircuit models in order to be able to link this sort of the static histological data to dynamic in vivo data. And then I'm thinking that the big brain or any other histological data can be a starting point for brain area specific microcircuit models. With that, I want to thank Shri, who is sort of our fMRI acquisition data analysis. Navona is in the audience. She will talk later about the claustrum, and she had a poster yesterday. You might be interested in looking at it. Sayan, Yueshin, Martin, they were all involved in the development of the hemodynamic models. And thank you for your attention.